Hello, my name is Emily RUEDA. I will talk about the origin of life. About 4 billion years, the Earth was a fireball with the surface covered only by a slight crust continually broken by the frequent fall of meteorites at that time till inhabited the solar system. In places where the crust had time to solidify and could potentially produce rainfall, forming ponds and lakes of liquid water that was not exactly but a mixture of water, ammonia, methane, acids, and salt suspensions. Later joined these atmospheric gases such as carbon monoxide and carbon dioxide and nitrogen. Shortly after, we no longer had a soup of atoms, but a soup of molecules quite complete. Successive boils, volcanic eruptions, lightning rain, bombardment of molecules that brought made from time to time. Many of these molecules were but destroyed, but also made to be formed by changing some more complex molecules. The energy was so great that simple substances tend to regroup as fast or faster than the destroyed completes. So, over millions of years, the stock was holding an increasing proportion of complete substance. Chains producing new molecules, millions of combinations each day worldwide. The unstable molecules were destroyed quickly. The most stable lingered longer. The simplest were used in experiments, one after another, day after day, year after year, millennium after millennium. But however complex these molecules were remained inert molecules. It took hundreds of millions of years of experiments to arise by chains of molecules capable of self-replication. For nearly 1,000 million years have developed a complex culture medium, and in that self-replicating molecules, the first soup had food and energy to play for hundreds of generations to cover the full extent of the seas. Millions, billions, trillions of experiments later, there was a molecule capable of surrounding a membrane leading to the first prokaryotic cell. Previously, it had emerged by chains molecules that were surrounded by a membrane. But the composition of the membrane was too strong, too impermeable, too fragile, or too whatever to make it useful. Those experiments failed. When one of the trillions of experiments succeeded, prokaryotes first appeared in history, more like a bacteria to a cell of our bodies that make up, but a living being able to read to more like a bacteria to its environment, protect from adverse conditions, feed and reproduce. Much more capable than the self-replicating molecules that populate the planet, the first prokaryotic cell is replicated again and again to produce the second baby boom in history. The expansion of life is not self-replicating molecules eliminate. Even today remain as virus and other prebiotic, but the planet was no longer the molecules, but the cells. Remain prokaryote cells, simple genetic material grouped in a membrane, such as what is now the nucleus of a cell. But how complex produce two opposing effects? One opposing effects. On the one hand, the cell was so complete the different parts of the molecule act under different conditions which made it more adaptable to their environment. On the other complexity, replication errors occurred more frequently in the case of molecules. Most of these failures cause the destruction of the cell, but other failures accounted for small changes in design. Sometimes the change was an advantage, sometimes it was detrimental change, and sometimes it was so totally neutral. Eventually came to be many different versions of the original cells, each with different probabilities of survival in different environments. At that time, there were millions of potential habitats. Some cells were able to survive in some that in others which led to the first specialization of life. Different habitats and different cells of the first painting the color picture of life on Earth. Cells were capable of making certain compounds and convert them into amino acids. Others could use the sun's energy to make sugars. Other cells, in short, could assemble amino acids to make proteins. The activity of each cell was unconscious and chaotic, but what each one was to go to places where they could survive better. The waste could be fed one to the other. It was inevitable that arose shortly after two or more groups of prokaryotes to form a colony with greater chance of survival than those with either alone. Were found thousands or millions of colonies, billions of experiments doomed to failure, membrane resulting in the first eukaryotic cells. Among all these strange and production of viable cells, which had no chance of survival, were destroyed immediately. but occasionally when there was a combination that had a better chance of survival than their counterparts. These cells compete with an advantage against their simpler predecessors, and in a few generations were able to eliminate their former supremacy. The reproduction of that first cell was still delicate, and errors occurred quite frequently. Sometimes a few components of the cell began to replicate earlier than others, leading to destruction of 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 the cell, and it went something completely different. Mutations almost always lead to the destruction of the cells, but some mutations were able to continue to survive and to reproduce by generating a different variety of the original cell. Sometimes there were beneficial mutations that made the progeny cells were better able to survive than their predecessors. Over time, cells formed complete unusual size. Some of our experiments have been found fossilized cells could be measured in centimeters. Thank you.